Hello and welcome everyone. Thanks for joining our webinar today. My name is Gurinder and I would like to kickstart our webinar with a poll question. So if you've joined the platform, you should be able to see a pop-up on your screen with the question. And I'd like to ask you, if you're new to lipid nanoparticles, liposome technology, or you're quite familiar with this and you've had some experience making or using LNPs for your application. So if you can, please answer the question on your screen, that'd be great. Okay, so I can see majority of you looks like are just starting to learn about it. That's great. 33% of you are considering using the technology. That's great too. And about 20% of you are already working with LNPs. So we've got a nice mix of people. That's really good to know, really insightful. So once again, welcome. We can get straight into our presentation today. So I'll share my screen. Okay, so I'm your host, Gurinder, and I'm going to discuss with you how best to control the synthesis of lipid nanoparticles. The topics, the titles I would like to cover in this session is basically introduce you to our team here at Dolomite, just to let you know what we do, what we have to offer. Talk about, obviously, the lipid nanoparticle space, introduce the microfluidic technology and why that's important when it comes to generating lipid nanoparticles. How best you can control their synthesis, what factors influence their final output, and we'll conclude and we'll take some questions in the end. So feel free to drop questions in the question box as you think of them, and we will be sure to address them at the end. Can you see the screen now?
Yes, thank you, fabulous.
Okay, great, thanks. So let's go straight to introducing our team at Dolomite Microfluidics. So we're here a team of chemists, engineer, biomedical scientists. We're a mix of different backgrounds. We focus on producing high-performance microfluidic platforms, which allow precision control over the size, shape, and structure of your final product. As well as LNPs, we also cover a wide range of different applications. We focus on a broad range of applications such as biomedical and pharmaceutical industries, and it can go beyond that as well. We supply standard products such as specialist chemicals and emulsion stabilizers. We advise customers on microfluidic setups and processes, especially if you are new to microfluidics or you're just getting to know the technology itself. We are there to help you along the way and figure out how best microfluidics can help in your laboratory setup and improve maybe even your industrial process as well. And if it's required, we can undertake some proof of principle projects as well. If you are unsure how exactly the technology will embed into your process, we are happy to do proof of principle studies as well for our customers ahead of any equipment purchase. So we do a wide range of different things, and we have a large focus on specific applications, one of which is lipid nanoparticles. So moving straight on to why lipid nanoparticles. All of you are here today because of one reason or another. You're interested in knowing more. One of the key examples we can take is what's going on in the world right now with the COVID example. It's the #1 problem. And aligned with that, what's exploded is the use of the nanoparticle space, and especially around liposomes and lipid nanoparticles has gained a lot of attention around the world with pharmaceutical and biotechnology companies. And the reason for this is, from the past, we've learned that the lifespan of any disease has directly been linked to the introduction of a vaccine. So as soon as a vaccine rollout takes place, you can see the lifespan of a disease is abruptly stopped. And that's what we've seen with COVID, where we've seen the fastest production or development of the vaccine for COVID-19, and it's by far broken the record in history for eradicating something that's causing such a worldwide pandemic. Most, if not all, vaccines require packaging before they're administered. So this is why LNPs have in the background also gained a lot of traction, has become a useful vehicle for delivering therapeutics. And that's because they are very, very biocompatible. And we can see here, this is even, in fact, this figure that you can see is actually taken pre-COVID. So I can only imagine right now the liposomes bar would be much, much higher than that. So the use of liposomes in any FDA-approved biotherapeutics, which are either in clinical trials or widely rolled out, is quite high. And just in May this year, the Google search also for lipid nanoparticles itself has grown from a couple of thousand searches to 15,000 searches per month. So we already know that there is a lot of traction, there's a lot of interest around the LNP space, and there are a lot of unmet needs in terms of the drug delivery and life sciences market, which we hopefully will try to address as well. So if you're new to LNPs and you kind of know what they are, essentially they're spherical nanoparticles composed of lipids with one or more bilayer. So here we've got just a single picture, a graphic of a liposome particle where you have your drug or biotherapeutic encapsulated in the lipid bilayer. And the advantages of using lipid nanoparticles is that they can encapsulate both hydrophilic and hydrophobic drugs. And that's quite useful because it offers that flexibility for you to be able to work with both types of API. They are non-toxic, flexible, biocompatible, and completely biodegradable, non-immunogenic for systemic and non-systemic administration. So they sort of mimic the biology of your internal tissues and membranes, which makes them really biocompatible and really flexible to be able to use. So hence why their popularity as well. Liposomes increase efficacy and the therapeutic index of your encapsulant. They reduce the toxicity that the encapsulating agent would cause if it was given as a raw product itself. They reduce exposure to sensitive tissues to the toxic API by protecting them, and also the site avoidance effect and flexibility to couple with site-specific ligands to achieve targeted delivery. And this is just some of the approved and marketed areas where they've used liposomes or lipid nanoparticles to deliver the API or the drug or the biotherapeutic. So this is just some of the examples and some of the industry that's currently approved and marketed. And this again is ever growing. So we're looking at the right space and the right thing. And if your application, if you're interested in something similar, and LNPs probably is the answer for you as well. So we know about why we need lipid nanoparticles, what they are. Now we need to understand how do you actually produce them? What is the technology that enables you as a user to be able to easily, go into a lab and be able to encapsulate your biotherapeutic? So one of the technologies that Dolomite is a leader in is microfluidic technology, and that's what we've leveraged here in terms of providing the best solution for LNP generation. Typically for other batch methods, you don't have your reproducibility, you have batch-to-batch variability, you have little control over the size itself. So microfluidic technology encompasses all of these and addresses the issues and offers the advantage over all of the conventional methods that you may be using. So the way lipid nanoparticles are produced in microfluidic technology is a process that we call nucleation and growth. So you see the diagram on your right-hand side. We've got two different phases which are being flow focused into a channel. And what happens here is we've got an organic phase which will contain your lipids. So it could be ethanol, it could be a solvent of your choice. And this is where you would have your lipids. So lipid, when you're making your formulation, you would normally dissolve it in a particular solvent or a mixture of different solvents. And this is introduced in let's say, the middle stream. And then you have your water phase, which is encompassing the middle stream, and this is where you would have your hydrophilic API or your biotherapeutic. So if you're working with, let's say, mRNA, you would usually put that in the water phase in some sort of buffer, for example. And you would flow focus these two into a stream, and it's during this process that the lipids will self-assemble and produce lipid nanoparticles. And because of this encompassing the lipid and the organic phase, the mRNA or your biotherapeutic also gets self-encapsulated when the self-assembly of lipid nanoparticles actually occurs. So they begin to assemble because there's a mixing going on, because there's miscibility between the two fluids. And the particle size itself can be controlled very easily by adjusting either the flow rate ratio or the total flow rate. When we talk about flow rate ratio, that means the flow rate at which the water phase or the lipid in ethanol organic phase is flowing at. So if they're both flowing at a 1-to-1 ratio, they could be producing a certain lipid nanoparticle size, let's say for 50 nanometers. And as soon as you increase the water phase to 2 to 1, you could increase it further to, let's say, 70 nanometers. So it allows you to have that kind of control over your lipid nanoparticle size. And similarly with the total flow rate, which can also influence your lipid nanoparticle size, but it can also increase the throughput. So what we mean by the total flow rate is the combined flow rate of your organic and your aqueous phase in total. So both of these parameters allow you to have control over the size and throughput. So the API, as I mentioned, can be brought in by the continuous phase, which is a hydrophilic water phase, or it can be also introduced in the organic phase, especially if your API is hydrophobic. And this can be encapsulated actively, or it can be encapsulated passively downstream as well. So the key advantages of this technology is that it offers high encapsulation efficiency because there's such a narrow path for the encapsulation to occur as opposed to batch methods where you use larger volumes. It has excellent PDI. So when we say PDI is the polydispersity index, which means how homogeneous your final product is. So the more homogeneous it is, the more predictable and downstream release kinetics can be monitored. It's reproducible, so every time you run the same experiment using the same flow parameters, you can be rest assured that your fluids are undergoing the same process. So hence it has really high reproducibility between different runs. It definitely uses low sample volume, so microfluidic technology itself is known to have the ability to really downstream screening and using low sample volumes to be able to optimize your process. So if you're at early stages and you do not want to waste your precious lipids or even your cargo that you're trying to encapsulate, microfluidic technology and that enables you to work with much smaller volumes, we're talking in the microliters range, as opposed to working with milliliters to liters. And it's also scalable. So whilst it enables you to work in smaller volume, it offers the opportunity to be able to scale your optimized formulation to be able to produce milliliters to liters at a time. So what you see on the screen at the bottom right is two different snapshots of microfluidic chips that we offer at Dolomite. And all you can see is basically the real-time production of lipid nanoparticles. And because they're nanoparticles, they're not visible to the naked eye, but the concept here is explained in the schematic above. And this is where you spot the two streams merging together. So the image on your left-hand side here is using a chip which we call the 3D flow focusing chip, which it has a 3D pore that you can see here, a little bit like a Coca-Cola bottle top. And then on the right, you can see the beginnings of a herringbone structure. So this is where we introduced turbulent mixing. So both of the chips can produce lipid nanoparticles, but they can give you slightly different results. So it's always interesting to be able to have that flexibility and control over how you produce your lipid nanoparticles. So since we are leaders in microfluidic technology, we have a dedicated system that we call the automated nanoparticle system that you see on your screen at the front of you. And what this enables you to do is, for users like yourself, is who don't necessarily know a lot about lipid nanoparticles or the production methods, and find that batch methods and conventional methods are quite cumbersome, laborious, and time-consuming. We have this system which allows you to use this technology with automation to produce a library of different samples. So if you're starting out, you want to try lots of different formulations, you want to try different combinations, you need to find out what's the most optimized size that works for you. And in order for you to be able to do that, you want to produce lots of little samples. So we've got a system here which comprises of some syringe pumps. So each syringe pair here is dedicated to a fluid stream. So we know that from our previous slide, we have the organic phase and we have the aqueous phase. So the aqueous phase is where we have our cargo, let's say mRNA, and the organic phase is where we have the lipids in our solvent. And then we also have a third syringe pair, which offers the ability for you to have a in-line dilution. So usually this helps the growth of your lipid nanoparticles and have a better homogeneous sample downstream. But the system allows you to have this as a completely optional step, so it's up to the user. When you're loading your cargo, your lipids in your solvent and your cargo in a buffer, for example, you would load them in this something we call the sample loops, which is connected to our automated sample valve. And what happens here is this syringe pumps allow you to release small snippets, depending on how you set up your experiment, of your sample towards the microfluidic chips. So they form that stream that we saw in the earlier slide and allow your system to collect a series of your different samples in this automated collector, which is fitted with a vial rack. So it will automatically move to the next stage, automating your sample collection. So I'll go on to explain a little bit more about that in the next slide. As part of this system, we offer different microfluidic chips that you see on the right-hand side. So at the top one, we see the T-junction, which is the flow impingement. So the T-junction I refer to as here. So all of these are glass microfluidic chips. They're very robust, they're usable, and you can autoclave them if you so wish. And so this particular chip offers two different ways to bring two fluids together, to basically mix two fluids together, which is very important when we are generating lipid nanoparticles. So you can either use one side, which is the T-junction flow impingement, which is usually what we use downstream, which is the second chip, when we want to use the dilution step. So if we want to use the dilution step, we usually introduce a dilution from this side of the chip. And then we've got the flow focusing of two fluids, which again is similar to the concept that was introduced in the previous slide. And this is that microfluidic chip that you also saw with the herringbone structures. So it allows a bit more turbulent flow mixing of the two fluids, as opposed to the flow focusing. Both of those would give you lipid nanoparticles, but it will vary the size slightly in either of those cases. So all three of these chips actually come as part of the system, so it allows you to have that flexibility to work with different types of mixing. And then lastly is also what you saw, the 3D pour, which was the Coca-Cola bottle top. And what this allows you to do is actually introduce the API as a middle phase, which allows it to compress against the organic phase. So as the mixing happens, there's better encapsulation efficiency. So they cater for different needs and they optimize depending on what your formulation is, but they all come part of the system itself. So how does the system work? This is just a very basic skeleton of the fluidic pathway of what the system is. So as I was saying, we've got the three different phases with the optional dilution, which happens in the second chip. As I've mentioned, it's usually in the T-junction here. But then you have your aqueous phase or your organic phase in the first two syringe pairs. And what they do is they draw the fluid from the driving fluid, which sits in the pressurized input store, that fills the syringes and it pushes the fluid, which is stored in the sample valve. You will load your sample into the sample valve, and usually this has a predefined volume, which is around 5 mils. And what that does is you load your sample for multiple different experiments, and dictated by your experimental table, it will get your repeats or your samples, so that it's only sending small volumes at a time towards the chip in order for you to generate lipid nanoparticles. And then the automated collector creates this library of different vials for you, the samples for you to test and analyze downstream. So the system automates a lot of this for you. You can dictate what it does by inputting the different parameters into the software that accompanies the hardware itself. If you want to know more, we're happy to share more information, and there are demo videos that we can share with you as well, so if you wanted to know more. So essentially, we make the generation of lipid nanoparticles easy by using a microfluidic platform, but also encompassing that in a system which is user-friendly and automates the process of optimization and generation of your formulation. So how do we control the synthesis of lipid nanoparticles? So this doesn't stop at just some of the ones that I've stated here. There are various different variables that can affect the synthesis of your lipid nanoparticles. So the first and the foremost, when you go into the lab and you think you want to encapsulate your biotherapeutic, a cargo that you have in mind, one of the fundamental important things that you want to think about is your formulation. And this then comes down to the user deciding what types of lipids they want to work with. And as you can see, depending on the complexity you want to work with, you can work with cationic lipids, helper lipids, pegylated lipids, and all of these and a combination of all of these will give you different results. And hence, once you've decided on the types of lipids you want to work with, the concentration what you use them in, and the ratio in which you use them in has a very important impact on your final lipid nanoparticle size, PDI, and then encapsulation efficiency, which is very, very important. So from a user perspective, that's something that you would need to figure out and you would need to think about. So once you've decided that, you can choose the chip that you want to use. And it's fine that if you want to screen against all the chip and mixing regimes that work best for you, that's absolutely fine. And then the flow rate ratio, the total flow rate that we spoke about, is very easy to control via the software. So you can just tell it, okay, I want to try to use a 5-to-1 ratio or a 1-to-1 ratio between my aqueous and organic phase. And as a screening process, this enables you to screen as many samples as you want, because it can produce very small volumes for you to analyze and quickly determine which way you would like to go. One of the things that we like to recommend is the use of DOE, design of experiments, which is sort of what is being dictated by the image here, is when you want to test different parameters and how that influences your final product, it's always a good idea to check at which level you want to test each of your variables. Other things which you are allowed to or can influence the final size and the product of your lipid nanoparticles is volume, such as your loading volume. How much sample are you using per generation, for example? And because we're using microfluidics and you're using smaller samples, the need to remove the front and the back part is also very important. So you're collecting the center hard cut, which can give you very accurate, reliable results each time, and as well as the optional dilution step as well. So there's a lot more variables which contribute to LNP synthesis and the output. So usually we would consider all of these and their influence as a response in size, PDI, and encapsulation efficiency. Although it's not limited to that, because in my next slide, you need to understand what's important to us about LNPs. So we know that we want them to be monodispersed, which means we want it to achieve low PDI. We want them to have size control. So we want to be able to produce, depending on what your end application is, maybe you want 20 nanometers, and maybe for another application, you might need 120 nanometers. So you want to be able to have that control over your lipid nanoparticle size. And you want to have consistency from optimization to manufacture. So you want to be able to produce smaller volumes when you're optimizing your formulation. But once you're happy with the end result, you want to be able to scale that on a bigger scale and be able to produce larger volumes. And as well as all of those things, we want to have encapsulation efficiency as high as possible, and obviously a controlled payload release. So downstream in your applications, when you're actually using that in vivo, for example, or doing some clinical trials, you want to be able to have that control in terms of payload release as well. So one of the things that I did as part of using this system is run a very simple formulation. So I used a 2-lipid formulation, you can see here, which formed the organic phase. So we've got phospholipid 90G, which is derived from soy lecithin, and DDAB, so a cationic lipid as well. So the combination of these two, this is a very basic, simple formulation in order to be able to show you how effective the system can be for your particular application. And then we've got the aqueous phase, which is using a buffer, which is just phosphate buffer saline. And the same phase was used for an in dilution as the aqueous phase. So this is buffer again. And then it's the flow rate ratio, which I decided I was going to vary across my different experiments. And the total flow rate I decided to fix. So here what I was testing is the effect of the flow rate ratio on the final lipid nanoparticle size. And so what you can see on the figure on the top right is how changing the flow rate ratio between the organic and the aqueous, including the dilution phase, has given me control over the size of the final lipid nanoparticles. You can see it goes anywhere between 20 to about 140 nanometers. So having that control just by manipulating one parameter is useful to know, and it's quite handy as well. So the second thing I wanted to look at is how I can achieve a low PDI. So if we look at the figure at the bottom here, is showing you a repeat of this particular sample here, and in 3 different repeats, which were done on completely 3 different experiments on different days. And what you can see how reproducible they are, but as well as that is the PDI is also very low. So we're looking at below 0.1, which is really good, because if you're looking to optimize and achieve a particular size and a PDI and encapsulation efficiency, in this case I haven't encapsulated anything, but if you wanted to achieve high encapsulation, it's these kind of experiments that you would need to do. So we can see that the system enables you to have very easy control over the flow rate ratio, which then controls your size and PDI. But on top of that, also achieve reproducible results. The reason why there's a little figure over here in the middle is because all of the figures that I'm going to show you are actually part of some posters that we are available for you to download as part of this webinar as well. So if you want to ponder over some of the details later on, you can download them from the webinar interface and look at them at your leisure. Just to reiterate as well, so the second figure that I showed you with the low PDI, these are just the representative Malvern master-size graphs. This is where you would analyze your lipid nanoparticle size and be able to extract the information in terms of the size and the PDI. And what you can see is 3 consistent peaks, and these are repeats of the same parameters, the same formulation, the same sample. And what you can see is how uniform they are, how tight the distribution is, and how that means that it's got a low PDI. So very reproducible results in all respects in terms of intensity, number, and volume. So that's quite useful to know. So the second bit we wanted to test is the influence of the total flow rate on the lipid nanoparticle size. So keeping the formulation the same, and as well as the organic and the aqueous and the dilution being included in them, I've changed the total flow rate, but kept the flow rate ratio the same. So in figure 2 here, the flow rate ratio was kept at 2:1:1, and in figure 3, it was kept consistent at 1.5, 1 and 1. So what you can see here is that despite changing the flow rate from 1 to 10 or 1 to 15, the final lipid nanoparticle size remained very, very consistent. So this means that you can increase your throughput without compromising on your final size of your lipid nanoparticle. And this means that you can get consistent results. And also, again, the PDI remains very consistent and reproducible as well. So again, the system allows you to have this type of flexibility to increase throughput and be able to screen multiple formulations in one time. So lastly, it's the figures that you can see here on the right-hand side. In this case, what the system allows you to do is, once you've made a library and optimized your formulation, and now you're ready to produce milliliters to liters of your sample, you can use the same platform to scale up. And this means that you can use it in continuous mode. So again, if you want more information, that's something that we can share with you and show you a demo, for example. And it's using the same interface, the same hardware, and the software that accompanies the system. And so what we're demonstrating here is by, again, using the same formulation and testing 3 different flow rate ratios and 2 different total flow rates, what we can see here is when we run it in protocol mode, this is when we're using smaller samples, we're generating a library of different formulations. We can compare that with if we were to then run it in continuous mode. So you can see in between all of these, scale up doesn't hugely affect the lipid nanoparticle size, and that goes for PDI as well. So there's no compromise in shifting from producing small volumes to producing continuously over time and producing upwards of milliliters to liters at a time. So in both cases, we're talking total flow rates of 3 ml per minute for figure 2, and figure 3, this was produced at 10 ml per minute. So this was running in a continuous mode at 10 ml per minute. And in each case, it's very, very reproducible. So in conclusion, we can say that lipid nanoparticles are a popular choice for targeted delivery of biotherapeutics. Microfluidic technology offers flexibility and control over lipid nanoparticle generation. The automated nanoparticle system offers a unique platform for optimization and continuous production of your lipid nanoparticles. And obviously the resulting lipid nanoparticles are very reproducible and homogeneous. There are many factors that can influence your final product. Technical factors such as mixing type and flow rate ratio, total flow rate, things like that can easily be controlled via the LNP system. And again, all of those graphs and any information around that is free for you to download as part of the webinar. We are happy to share some more live demos and talk more about with you about your particular application, especially if you're interested in microfluidics and how we can bring the 2 fluids together and help improve and optimize your LNP generation for your particular application. So thank you for listening, and I'm happy to take any questions as well. But if you think of something after today and you want to know more, do feel free to send us an email and we'd be happy to get back to you. So we're going to allow a few minutes. I'm just going to open up the questions. Okay, so is miscibility of solvents required, or is it also possible to use a mix of miscible and slightly soluble solvents for lipids? Yes, exactly. So there's some miscibility is required, because in order for the lipid nanoparticles to self-assemble, we require the two phases to mix and be able to have some miscibilities to allow your cargo to then get encapsulated. If there was no miscibility at all, your cargo or your API will not be able to get encapsulated, and your lipid nanoparticles will not be able to assemble. So usually the most commonly used solvents are ethanol and the likes of ethanol, or mixtures of these types of solvents, which are somewhat miscible in an aqueous phase. And they work really well as well. Okay, how do I choose from 3 chips which one to use for my LNP? That's a very good question. And that's definitely something we can have a whole different session about. Essentially, it's the way we mix 2 fluids together can influence the final size of your lipid nanoparticle. Usually what we found is from flow focusing to turbulent mixing, there's an increase in the final lipid nanoparticle size, essentially. So it enables you to push the boundary in terms of how large you can make your lipid nanoparticles. I think the question is how the temperature impacts on the production of LNPs. So at the moment with this particular system, we don't have temperature control, but it's definitely something that we're looking into and something we would look to incorporate in let's say, the next generation. But because our chips are glass, they can very easily be mounted on, let's say, a hot plate, if that's something that you really need as part of your system. And that way you can actually monitor the temperature during the production of your lipid nanoparticles. So at the moment, it doesn't come as part of the system. But as I mentioned, because the chips are made of glass and they are quite robust in terms of how they can withstand high temperatures, they can easily be placed on a hot plate. So that's not a problem, but that's something that would be an add-on feature. Okay, once the lipid nanoparticle solution was obtained, you have to remove the ethanol and or concentrate. Yeah, how do you do that? So there's a lot of downstream processes already available, which are sort of off the shelf. This is usually done by dialysis, membrane dialysis. You can also purchase some membranes as well from online, and they are quite compatible with your ethanol, with the solvent of your choice, like ethanol. And these are very easy to do in a typical lab, and usually done downstream. It's not something that's done part of the system, but there are lots of off-the-shelf solutions available. So once you start generating lipid nanoparticles, it's very easy to implement that afterwards. Our need is to make polymeric micelles. How would the dolomite system work with this? So very, very similar. The technology itself isn't limited to lipid nanoparticles itself. The same platform can be used to make polymeric nanoparticles as well as micelles. The concept remains the same. You would have the 2 phases that you load in the 2 automated sample valves. You would bring them together using one of our microfluidic chips, and the end results would be your micelles, and this can easily be analyzed downstream. So although the system was developed with keeping lipid nanoparticles in mind, it is by no means limited to use with lipid nanoparticles. You can use it with polymeric micelles or nanoparticles as well. So is it possible to achieve synthesis of liposomes and lipid nanoparticles using the same chip, materials and settings? If so, what discriminates the formation of the first instead of the second? So usually it is any of those formulation variables that can affect how the self-assembly occurs, as well as the way we mix the 2 fluids. The only way of really determining whether you're making liposomes or lipid nanoparticles is to be able to do some image analysis. So whether you're using a TEM, SEM, that way you would be able to determine whether you're making one or the other. But going with the system as running it from one way to another is very difficult for you to know from the point you collect if you've made one or the other. So that requires a lot of downstream analysis in terms of morphological analysis in order to know what part of your formulation is influencing it one way or the other. Okay, so do we have a complete comparison between conventional stirring mixing method and microfluidic system? May the particle size tunability is achievable by conventional mixing as well in that range? I think I get the first part of this question. So yes, there's a complete comparison definitely available in terms of comparing microfluidic systems to conventional stirring methods, and you will find the conventional stirring methods do lack in terms of how much you can tune particle size. There's limited, like you're limited by the amount that you use, so the volume that you have, and also the mixing process itself introduces a lot of variation in the batch process, as opposed to when you use microfluidics methods, which are very, very reproducible. So there is a lot out there in literature as well, which you can have a look at, but microfluidic technique by far produces much better lipid nanoparticles as opposed to stirring methods. I am just going to take one more question because we are running out of time. So how about fouling during continuous production? What are typical production times in the continuous mode? So in terms of fouling, depends on how long you want to run it for. Most of our components with the system are very robust and reusable. So although in long term they are consumable and something that you can very easily change in terms of the chips themselves, the tubing and or the syringes, but usually they are very, very robust and they can last a very, very long time. And it comes down to maintenance as well as, robust cleaning regimes that need to happen after every process that's you run with the system. So, and in terms of typical production times in continuous mode, you can achieve upwards of 20 ml per minute in continuous mode. So depending on what is you're trying to achieve downstream, you should be able to achieve that very easily. But those are the type of total flow rates we're talking about. So I think we are, what I will do is the questions keep coming. So what I will do is reply to all of you one way or another, either via email. But thank you so much for all the lovely questions and attending the webinar today. Really appreciate your time. But definitely do take the handouts that are available. If you do want to know more about the system, how it works, or how the chips work better for you, please feel free to drop us an email and we will be sure to get back to you. So thank you ever so much for joining, and hopefully we'll see you again soon next time. Thank you.